All right, AP Bio, welcome to the Cell Communication PowerPoint. This is the one we're working on for the next 2 weeks. So there's like 39 slides that we're going to kind of move. So we're talking about Unit 4 in Cell Communication. Like, I'm going to ask you these questions in class. Why do you communicate? How do you communicate? Right? How do you think cells communicate? And do you think bacteria communicate and explain? And then we're also going to talk about comparing the structure and functions of receptor proteins. We'll talk about what is a second messenger and what are the possible responsibles to signal transduction. And so we talk about cell communication. You'll watch a video in class. I'm going to skip through that. We'll skip through these questions. It's about cell signaling. So animal cells, like the ones in your body, communicate by direct contact, which is a gap junction. They secrete local regulators, things like growth factors and neurotransmitters, so like hormones, if we're talking about endocrine system, or excuse me, long distance is hormones, local regulators are neurotransmitters. Excuse me about that. So here's just an image of local signaling, and then you have paracrine, synaptic, and long distance. But here, this is the main point of this part, is that there are 3 stages of cell signaling. There's reception, transduction, and response. Reception is the detection of that signal molecule, the ligand. You have a transduction, which converts that signal to a form that can bring about a response, and then you have the response. And so we're going to be labeling this picture in class, and you'll have an FRQ over it. So talking about the reception, it's the binding between a signal molecule and a receptor. It's highly specific. Think of lock and key enzyme substrate. And we talk about receptors, we talk about a plasma membrane receptor and an intracellular receptor. So plasma membrane receptors are water soluble. And so what these mean is that the receptor's on the plasma membrane. And so there's a charge. Intracellular, hydrophobic, or small ligands, they can pass right through, and things like testosterone or nitric oxide. So the ligand binds receptor protein. The protein will change shape. It'll have a conformational change, and then that'll initiate the transduction signal. And so you've got the G protein coupled receptor, tyrosine kinase, and ligand-gated ion channels. So there's your G protein coupled receptor. Again, I'm going to kind of move through these. You can pause them if you want. You may just want to pause here and jot in the chart. But the G protein coupled receptor, the GPCR, there's basically 7 transmembrane segments in a membrane. And the G protein plus the GTP, remember ATP, well, GTP, activate an enzyme that leads to a cell response. So G protein coupled receptor is one where there's a special protein, there's a special G protein, right? You can see that in purple. The signaling molecule attaches it, and GDP or GTP, right? It phosphorylates and it provides the energy for that G protein to become active, and it moves across. And we'll spend more time on that. Receptor tyrosine kinases, again, we know these are enzymes, and so think of kinase, think of kinesiology and movement. And so again, if you've watched the AP videos, I talk about this, but there are, again, the signaling molecule attaches, cause a conformational change, or what we'll call a dimer form, and then that dimer will activate a receptor again, and then that receptor there in question number 4 will then activate cell response 1 and 2. Again, you're not going to have to memorize all of these steps, just kind of knowing that there's different ways for receptor membranes to function. So in a tyrosine kinase attaches a phosphate to a tyrosine, so it phosphorylates it, and that can activate multiple cellular responses at once. Ligand-gated ion channel, again, there's your signaling molecule. It attaches to that gate, which is closed. When it attaches, it opens. Then the materials can move through, cause a cell response, and then the gate will close. And so ligand-gated ion channels signal on a receptor changes shape, regulates flow, especially calcium and sodium. So here's the chart completed. If you want to pause it, you can. So transduction, this is the cascade. So if you think of a waterfall cascading, this is, I'm trying to do 2 things at once here, so give me a second so I can click on this. It's these molecular interactions that relay signal from receptor to target molecule. And so it's a cascade, and it moves it along. So protein kinases are enzymes that phosphorylate and activate proteins, and they lead to a phosphorylation cascade, which can amplify a signal. And so if you look at this image here, there's your signaling molecule attached to the receptor. Now, if you notice, the receptor or the signaling molecule is not going into the cell. That reception will then activate a relay molecule. So inactive protein kinase 1 becomes active. Then active protein kinase 1 can activate 2. 2 activates, so 2 becomes active, activates 3. Activate 3 becomes active. And then active protein kinase 3 can then inactivate the protein, activate it, lead to cell response. So going from 1 to 3, that is a phosphorylation cascade. So there are things called second messengers, and these are small non-protein molecules or ions that can relay signals inside. So you have cyclic AMP or CAMP, calcium ions if you talk about muscle contractions, and then IP3, inositol triphosphate. But again, these are all just examples of second messengers. But CAMP is the one, it's cyclic, how you want to say that, adenosine or adenosine monophosphate. And if you can see there in the picture, you have your adenyl cyclase, right? The enzyme, once it's activated, then ATP will activate CAMP, becomes a secondary messenger, protein kinase A to cell response. So let's talk about response. So response, you regulate protein synthesis by turning on and off genes in the nucleus. That's called gene expression. And what it does, it'll regulate the activity of proteins in cytoplasm. And so what'll happen is the response, usually it tends to be like transcription, or the response will be producing a gene. So let's talk about types of hormones real quick. Steroid hormones, we're going to fill this in class. But again, these are ones that can pass right through. So there's some examples, progesterone, testosterone, estrogen. So there's some examples. And here's the protein hormones. And again, the picture got cut up. I'll try to fix it. But these ones are hydrophilic, so they have to have a receptor protein. And so here's some example proteins. So we have ADH, antidiuretic hormone, oxytocin, insulin, glucagon, ACTH, parathyroid hormone, follicle stimulating hormone there, FSH, luteinizing hormone, and then TSH. So some example of cell communication, but what we're also going to be talking about, and this is what you're going to be doing the activity for, you're going to be talking about signal transduction problems or defects. So things like diabetes, cholera, autoimmune disease, cancer, neurotoxins, poisons, and pesticides all can disrupt the signal transduction pathway, and things like drugs, so anesthetics, antihistamines, blood pressure meds. And so there's cholera. You can pause. I'm not going to expect you to know the specifics of each of these. Viagra, how that works. And then, of course, apoptosis is cell suicide. Essentially, it's where the cells break down, and that's all due to cell communication. And so what will happen is the cell will be dismantled and digested, but it's triggered by signals that activate a cascade of what we call suicide proteins called caspase. And so why? Just basically, if a cell has an issue, it'll protect the neighboring cells. And they think that this apoptosis and this caspase may be involved in Parkinson's and Alzheimer's. So here's an example of apoptosis of human white blood cell. The left is normal, the right is undergoing apoptosis. So you can see how it's kind of being broken down. And then here's some apoptosis during pod development in mice. So again, I ran through that quickly, but I just want, these are the notes we're doing in class. So you can kind of watch it ahead of time. I'm not going to be able to mark up the screen, but if you kind of want to watch and kind of get a quick preview. So that's all I have for you. I reduced it down to 7 minutes. We'll also be doing immune system and nervous system and endocrine system. We do cell communication, so we're just doing like just the basics of that. And then once that's wrapped up, the next part of Unit 4 will be the cell cycle and mitosis, which I think we should be able to move through pretty quickly. We'll see. Anyways, hope you found this helpful. Again, if you got any questions, let me know. This is me signing off from room 102 at Tiffin Columbian High School on Tiffin's beautiful South Side, and I will see you in class.